Hi, my name is Dora, and today I'll be presenting my research from my time in the Lindbergh lab, focusing on investigating soluble joint tissue signaling between 2D muscle cells and 3D cartilage spheroids. Now, to give a little bit of background, the joint space is very complex in that it allows for interaction between many different tissue types. And here in the Lindbergh lab, we have a focus on studying post-traumatic osteoarthritis, also known as PTOA. This is the most common disorder of the joint, and it's characterized by cartilage degradation caused by a traumatic injury that then leads to chronic pain and decreased mobility. Although it is the most common chronic disorder, still not a lot is understood about this condition. So we utilize 3D cartilage spheroids in our lab to help better research and understand this disease's progression and its mechanisms. So we begin with articular chondrocytes that are cultured until confluency, and they are then spun down and centrifuged to form a chondrocyte spheroid or a pellet. They are then introduced to TNF-β to form healthy cartilage tissue, and to simulate the progression of PTOA, we then introduce these pellets to pro-inflammatory cytokines, TNF-α and interleukin-1β. Now, what is healthy and diseased cartilage tissue defined by? Well, cartilage tissue has previously been defined by the presence of glycosaminoglycans, also known as GAG. GAG is a key component in cartilage, and it's responsible for cartilage, its ability to compress and its stress endurance capabilities. Now, when we analyze our 3D cartilage spheroids, we find that we are able to create these spheroids with the presence of GAG and then show that post-cytokine exposure, so after introducing them to TNF-α and interleukin-1β, we show that there is a degradation of this GAG quantity, and this is done through histological analysis and also showed through our quantified GAG per DNA data as well. So during our baseline, during the beginning of the project, there is a higher amount of GAG per DNA, and there is a significant decrease in this GAG per DNA after the pellet's exposure to our pro-inflammatory cytokines. Now, going back to the background, earlier I said that the joint space is very complex and there are many different tissue types that interact with one another within the joint space. So a lot of previous literature has focused on just cartilage and bone mainly. So we thought, what about muscle? What about this other major tissue type within the joint capsule that is important for the interactions of the joint? When looking at this model, however, muscle and cartilage are often not touching each other. They're not in direct contact. So how do we study these two tissue types in a way that is physiologically relevant? We decided to utilize the idea of a transwell indirect coculture process, where our 3D chondrocyte spheroid will sit within a transwell insert, and a 2D muscle cell layer on the bottom of the plate will allow for soluble signals to pass through our transwell insert. And so the two cell types will communicate with one another. And this model is much more physiologically relevant since it demonstrates the communication between these two tissue types without them being in direct contact with one another. Coculture often has these two different cell types together in a mixed population. And so in this way, we are able to study it in a more physiologically relevant way. But with these, before we can coculture in this transwell insert, there are two separate concepts we must be aware of and think about. First off is the insert design. Are we able to design and 3D print a reusable well insert that is compatible with our 96 well plates that we use in our cell culture process? And our second idea that we must think about is the media composition. Will chondrogenic media affect muscle differentiation processes? And so if we are co-culturing muscle cells and chondrocytes, how will they interact with each other, and will our media composition be detrimental to either of those cell populations? So focusing first on the transwell insert design, this is a CAD model of the insert design that I designed myself using the Autodesk Inventor program. After optimizing this model with measurements and doing some prototype prints, we then utilized an SLA printer, also known as a stereolithography printer, to print our design. Now, an SLA printer utilizes a resin reservoir, and so a platform is dunked into the resin reservoir, and a laser permeabilizes the resin layer by layer, and so the platform lifts up, allows excess resin to drip, and then is dunked back in again, just like this short video clip showing a SLA printer in action. And so we decided to utilize the Dental LT Clear V2 resin specifically for its biocompatibility and its autoclavability. So we are going to be able to reuse this resin per its specifications, and it is also biocompatible, meaning that we are able to use it with cells and it's not biotoxic. And so this is a picture of one of our prototype prints, one of our first prints that was successful. And the resolution of an SLA printer is quite clear. So you can see that these holes can get up to 400 microns in diameter. Focusing then on the media composition, we tested our media by culturing chondrocyte media and muscle cell line media on primary muscle cells. And so we used primary muscle cells taken from surgeries at Slocum Orthopedics, so they are actually patient samples, and we cultured them alongside a C2, C12 myoblast cell line. And this is a well understood, well researched cell line, and so we'll use this as a control to our primary muscle cells to see how these two different cell types interact to chondrocyte and myocyte media. And so once these two cell types, and these are cultured separately, so they are not co-cultured, just to give a little bit of more information there. And so once these two cell types were cultured to confluency, we then tested the effects of different media compositions. And so just to focus a little bit on what confluency means here, confluency is when the muscles form a monolayer on the bottom of the plate. And so it means that the cell population is high enough for us to then test. And we made sure that we plated each of these wells with the same number of cells to make sure we account for that. We will also account for that in our analysis as well. And so once these cells were cultured to confluency, we then tested its effects on 4 different media types. So we tested a group of muscle expansion alone, a group of muscle differentiation media alone, and then we had 2 groups that were a mixture of different media compositions. So we had a group with 1-to-1 chondrogenic and muscle expansion media, and then another group with 1-to-1 chondrogenic and muscle differentiation media to see its effects on the muscle cell growth, proliferation, and possibly differentiation as well. And during this process, we actually also collected conditioned media for future experiments, which I will touch on later in our future steps. And so after the media was switched to the 4 different treatment groups, we made sure to collect metabolic activity analyses on the groups during different time points during the experiment to make sure we kept track of how active these cells were in these different media types. And so looking at the timeline, we plated these cells, and after 3 days, that's when the experiment started on day 0, we switched the medias and changed medias on day 1, and then every 2 days after that. So we changed medias to their different treatment groups on day 0, Then after that, changed medias on days 1, 3, 5, and 7, and made sure to collect metabolic analysis time points throughout these media changes, and then collected the cells at the final time point on day 0. So looking at the metabolic activity analysis data, it shows that the C2C12s did not relatively change much to from their day 1 activity, metabolic activity levels to day 0. And this graph is the numbers are normalized to baseline levels of day 0. So on days 0 and days 1, they are normalized, and then we're comparing that normalized data of day 1, which is in this blue here, and day 7, which is in the purple. And so there's not a lot of statistical difference between these two time points for the C2, C12 cell line. But when we compare this data to the primary muscle cell data, there is a lot more statistical significance of metabolic activity between days 1 and 7, especially with the expansion media alone. That treatment group had a very significant difference in metabolic activity. So this data proves that chondrogenic media is not detrimental to primary muscle cell growth, and if anything may show to increase their metabolic activity. And so with this, our next steps would be to use the conditioned media that I said we collected earlier and use that on and expose them to our cartilage spheroids to see if the secreted proteins from the muscle cells have any effect on our cartilage spheroids. And we will also need to test the sterility of our inserts to see if those 3D printed inserts I created and 3D printed earlier, if they are able to hold sterile when we are using them in cell culture, and to prove that they are biocompatible. And so I would like to thank everyone who has helped me throughout my research journey, and I would like to thank the WUSI Human Performance Alliance for funding, and also the Knight Campus Undergraduate Scholars Program for helping funding my research. And thank you so much for listening.